Typing up an assignment is a simple task for most of us. But how many of you have ever stopped to consider the physiological processes that are occurring? Our ability to perform these tasks, among others, revolves around the structure and function of the neuromuscular junction, a key interface in our body. But first, what is the neuromuscular junction? How does it work to contract muscle? And why do those with neuromuscular diseases, such as myasthenia gravis, suffer from muscular weakness? This video tutorial will explain how the neuromuscular junction works in healthy individuals, and will also explore what happens when this junction malfunctions. The neuromuscular junction is the crucial meeting point between the nervous system and the musculoskeletal system. This communication allows normal bodily movement to occur. When we decide to move our body, our brain sends a neural signal to our skeletal muscle. The neural signal, or action potential, is propagated down motor neurons. Motor neurons are a collection of nerve cells that carry neurogenic signals from the spinal cord to the muscle you are trying to stimulate. Before the muscle contracts, the nerve signal reaches the neuromuscular junction. For us to understand the function of the neuromuscular junction, we should get to know the key structural components. It is composed of the presynaptic nerve terminal, the synaptic cleft, and the postsynaptic muscle membrane. Fun fact, the synaptic cleft, the space between the presynaptic nerve terminal and the postsynaptic muscle membrane, is 400 times smaller than the width of a red blood cell. Key processes to understand are the movement of acetylcholine and calcium ions. Now let's get into the nitty-gritty of the neuromuscular junction. Once the action potential reaches the presynaptic nerve terminal, the presynaptic membrane is depolarized by the influx of sodium ions. As a result of the change in membrane potential, voltage-gated calcium channels open and calcium ions flow down their concentration gradient from the extracellular fluid into the neuron. The influx of these calcium ions from the extracellular fluid triggers the exocytosis of neurotransmitters called acetylcholine. Calcium ions bind to the synaptic vesicles. This, in turn, causes synaptic vesicle exocytosis to occur. Exocytosis is a Greek word where exo means outside and cytos means hollow vessel. We see this as exocytosis is a process where a vesicle or a hollow vessel merges with the cell membrane, allowing the release of acetylcholine into the synaptic cleft. Acetylcholine crosses this gap and activates the nicotinic acetylcholine receptors located on the postsynaptic muscle membrane. These nicotinic acetylcholine receptors are non-selective cation channels that allow potassium efflux and sodium influx. However, the net positive charge entering the cell depolarizes the sarcolemma, which is the muscle cell membrane, and results in the end-plate potential. The depolarizing current of the end-plate potential opens voltage-gated sodium channels that allow rapid sodium influx, which triggers an action potential. As the muscle action potential propagates along the sarcolemma and down the transverse tubules, also called T-tubules, it stimulates the release of calcium from the lateral sacs of the sarcoplasmic reticulum. It is this increase in intracellular calcium that facilitates muscle contraction. Once the brain decides to stop muscle contraction, what happens at the neuromuscular junction for this to occur? Since acetylcholine is integral for initiating the contractile response, it seems intuitive that removing this chemical from the synaptic cleft will cease contraction. Acetylcholine esterase, both in the synaptic cleft and bound to the postsynaptic membrane, is an enzyme that breaks down acetylcholine in the synaptic cleft into choline and acetic acid. After this acetylcholine is broken down, reuptake of the choline in the presynaptic neuron occurs via choline transporters and allows the choline to be reused in the synthesis of new acetylcholine. Without acetylcholine, the receptors on the postsynaptic membrane are allowed to return to a closed resting state and an action potential cannot be propagated. However, in myasthenia gravis, the communication that occurs at the neuromuscular junction is impeded. This leads to skeletal muscle weakness and later fatigue. In patients with myasthenia gravis, the nicotinic acetylcholine receptors are blocked, altered, or degraded by the body's immune system. This results in fewer fully functional receptors. Let's apply our understandings of the intricacies of the neuromuscular junction to a foreign context. How would you treat this disease? Let's look at some options. We could A. Increase the acetylcholinesterase concentration. B. Increase the sodium concentration via sodium supplements. Or C. Use acetylcholinesterase inhibitors. Let's think through these answers. Acetylcholinesterase degrades acetylcholine in the synaptic cleft. This would lead to less receptor activation and thus amplifies the symptoms of myasthenia gravis. Let's evaluate option B. In option B, we know that sodium is necessary for action potential propagation. However, we also know that sodium cannot enter and depolarize the muscle cell unless the receptors are functional, activated, and open. It appears that option C may be the correct solution. By blocking acetylcholinesterase, acetylcholine is present in the synaptic cleft for a longer period of time, meaning that there is a greater probability that the receptors are activated. Hence, we see that the main way to treat myasthenia gravis is to inhibit acetylcholinesterase with drugs such as neostigmine and pyridostigmine. Thus, option C is the correct answer. So in summary, the neuromuscular junction facilitates communication between neurons and muscle cells. Action potentials are propagated down neurons to the presynaptic terminal. Voltage-gated sodium channels open, allowing sodium to enter and depolarize the presynaptic nerve cell. The voltage-gated calcium channels open and allow calcium to enter and trigger synaptic vesicle exocytosis. Acetylcholine is then released into the synaptic cleft and binds to the nicotinic acetylcholine receptors on the postsynaptic muscle membrane. Sodium ions enter the muscle cell and propagate the action potential along the sarcolemma, down the T-tubules, and thus stimulate muscle contraction. We've also learned that the symptoms of myasthenia gravis are caused by impaired receptor functionality on the muscle cell membranes due to an autoimmune attack, but this disease can be treated with acetylcholinesterase inhibitors. So in this video, we have covered the neuromuscular junction, structure and function, exocytosis of acetylcholine, action potential propagation stimulating muscle contraction, and what happens when things go wrong. For more information and resources to consolidate your knowledge, follow the links in the description box. Thanks for watching.